Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about cytoskeleton and cell motility. We want to survey the structure, the protein composition, the functions of the complex network of cytoplasmic filaments and tubules known collectively as the cytoskeleton. Thank you. Here we can see body whorls indicating the nervous system, which has a lot of cytoskeletal components to be able to transmit messages from the brain to the rest of the body. Now, the cytoskeleton, you must perform certain tasks, and one is for structural framework. So you have to have a structural framework in the cell. It has to maintain the shape. It has to move out pseudopodia to be able to move the shape, change the shape associated with movement or with phagocytosis, endocytosis, and phagocytosis of the thing for the cell to stay alive and to maybe do its function if it's a macrophage, and stability of attachments should the cell attach and move to another cell. So cells must perform certain tasks that require some kind of contractile machinery. Transport of vessels is one thing. Movement of organelles, movement of chromosomes, division of the cytoplasm. You have cytoplasm pinching off at the cell division. Specialization of cell surfaces. And we see some of those pseudopodia projecting here and these microspikes for it to interact with the environment. And also cell motility. These are some of the characteristics of the cells that must be performed requiring cytoskeletal components. And cytoskeletal components are of three types. You've got microtubules, which are the longer ones, which is basically a tube. It's about 25 nanometers. Microfilaments is the smallest one. That's the actin filaments, and that's 6 nanometers. And then you have something in the middle, intermediate filaments, and that's about 10 nanometers. And we see all three of those together as all of them are proteins and stained here with chromatic blue. These are known as non-membranous organelles because they are not associated with membranes. They're just in the cytosol. Microtubules is one of the ones that we want to look at. It's a big one. It's composed of tubulin, which is highly conserved over the millennia. And here we see a tubulin molecule, which has an alpha and beta component to it. And these are lined up and make these protofilaments, and then a certain number of protofilaments go together to make a microtubule, which has an opening in the inner center. So there's 13 of those protofilaments that come around through there, and each is composed of alpha and beta dimer. These ones are labile microtubules in that they associate and disassociate and perform their functions by whether or not it's associated intact or it's broken up. Microtubule-associated proteins, MAPs, are one that stabilize microtubules, and here we see microtubules up through here with the protofilaments across sections of them, but sometimes we'll see proteins associated with those microtubules, and that will give it stability, like in a cilium or like in a centriole. And so microtubule ultrastructure, as we can see, you have this alpha and beta tubulin that is coming together to make a tube, and so we see the alpha beta dimer. Each one of these are dimers, and each of these go together to make 13 cytoplasmic one microtube of 13 protofilaments. You can also have these in a doublet form, and here we can see doublets. This is in a sperm tail, but a series of doublets with the dining arms, and the dining arms interact with the other doublets for the sliding to occur. So it can go singles, doubles, or you can have triplets. It could be triplets here, and in the doubles and the triplets, you don't have 26. Some of them are shared in the middle. So 13 here, 13 there, but some of them are shared in the middle. And here we can see the triplet microtubules here and here and here with no central pair. When you have doublets, you have a central pair, but you don't in the centriole. Basal bodies of cilia at the bottom, at the top of the cell, where you have cilia projecting, you have triplet microtubules. So here we see a cytoplasmic microtubule. There's one here. You can see these long projections, and this to show you the stable and labile microtubules. There's a nucleus here. There's the limits of the cell, and this one just shows you the stable ones. So certain ones are stable because they have MAPs, or microtubule-associated proteins, with them, but the labile ones are ones that are breaking down and building up to perform the functions of the cell. So we see microtubules here, here, and here. The function of microtubule is to direct the contractile force. So they use actin as the main contractile protein, but it's directed by microtubules. So they direct where the cytoplasm will go. And here we can see another thing that they do. They transport vesicles as move organelles along the way. And here you can see, this is from the internet, where it shows a vesicle being transported along a microtubule. And so you move vesicles, you organize the Golgi, Ruffy mitochondria, you also separate chromosomes. Those are the different components of the functions of the microtubules. And here we see microtubules. There's a nucleus over here, a centriole sending out, radiating out labile microtubules, and here in this case, where we see a cell, this is a T cell that kills maybe a cancerous cells, and you can see where the centriole moves here, and microtubules are radiating from that, directing vesicles into here. This is a nerve here, and so it shows you the terminal of a nerve. So microtubules are associated with anterograde and retrograde transmission of nerve cells. And here we can see the same cell. This is a nucleus, and this one shows you the microtubules, where the microtubules are in high concentration in the cell, and where mitochondria are. And here we can see mitochondria. In other words, the microtubules help direct the location of the mitochondria. Here's endoplasmic reticulum. The nucleus would be in through there, but endoplasmic reticulum branching and anastomosing, and it's the same cell labeled for microtubules. So you can see that the microtubules are pretty much the same as where you have endoplasmic reticulum associated with it. Here again we see microtubules shown here as a nucleus, and this is showing the Golgi apparatus with microtubules present. And if you know that the Golgi apparatus is associated with close to the nucleus. That's where you usually find it. But if you treat the cell to get rid of the microtubules, you can see the Golgi apparatus is dispersed. In other words, it's held in check in this location by the microtubule. So organization of Golgi, as we see here, ER, as we saw a while ago, or mitochondria, is one of the functions of microtubules. So where microtubules are found, you're going to find them directing these other organelles as well. Now in the centriole, we see the centriole right here. There's a nucleus, and the nucleus here of the cell. And so this is where the cell was treated with colchicine and then allowed to recover. And here you can see that microtubules are radiating from the centriole. So you get regrowth from the centriole. So a centriole is a duplex in the center of the cell, is composed of triplet, 9 triplet microtubules. A deposome is a pair of centrioles, and centrioles are self-duplicating. They develop from pre-existing precentrials is where they come from. Here we see centrials here and here, and so we see where the chromosomes are pulled apart. These are labile microtubules, as we can see. You can see that coming from the centrioles are the labile microtubules that project out, that connect with the nucleus, with the chromosomes in the nucleus, and actually the nuclear envelope been removed at that point in time. So centrioles come from previous centrioles. In the case of cilia, flagella, basal bodies at the bottom of ciliated cells is where you see root-like devices that the cilia is produced from, and also it's anchored there. The functions organized microtubules is to pull chromosomes apart, is one of the things that it does. Here we see the centriolar triplets, centriole here, and then more centrioles coming from, you get some signal from the nucleus and some products that are coming here. Finally, you get the production of new centrioles, which are actually formed of basal bodies of these, but they're from like the procentrioles. So here we see centriole here, proximal and distal centriole that are located there. This would be the nucleus of a sperm in through there. Actually, this would be the cytoplasm. The nucleus is not attached to the tail yet. And here we see the centriole giving rise to the developing tail. And here in the ear we can see the triplet microtubules. So 1, 2, 3, 1, 2, 3 microtubules. And actually there's an axoneme in here with a central pair. And so in the centriole itself, you have the triplets. In the axoneme coming out from there, you have doublet microtubules. And we can see that here. Here's the zona occludens at the apex of the cell. This is one cell, another cell. You see some microvilli projecting in through there. It's branched. But we also see cilium, and these are the basal bodies. And through here, you can see the triplet microtubules in through there, as opposed to here we see the typical axoneme, which are the doublets. So we have doublets over here, triplets down here, and these are the basal bodies that support the developing cilium, and the cilium itself actually grew out of the centriole. So in the axoneme of a cilium or flagellum, we see doublets, central pair, we see radial spokes, we see dining arms. These are the maps that makes these microtubules stable. So when they first started discovering microtubules, they found them in the stables case in the beginning, and it was on until they got the glutaraldehyde and some other fixatives that they were able to see the labile microtubules. Usually the process of fixing and killing the cells eliminates the microtubules that are in there. So you don't depolymerize stable microtubules because they're attached to the microtubules. So the microtubules here we see the dining arms, which has ATPase activity, and it interacts with the adjacent one in the case of sliding back and forth. Also has a central pair of microtubules as well. So we can see there these are stable. They have dining arms, as we said. The lateral appendices, this is ATPase activity associated with the cell moving, because a doublet will move in relation to another, and that makes the tail swing back and forth, as we see. So if we look at different portions of the tail of a sperm, this happened to be from a horse, we see the mitochondria around the outside of the middle piece, dense fibers, and then we see the axoneme inside there. So here's the axoneme. Here you see the dining arms, doublets, and if you get to the very end piece, each one of these doublets makes 2 microtubules. This one we can see doublets and singlets, and so there should be 20 singlets at the very end. That is 9 times 2 is 18 plus 2 is 20. And here we can see the different components of the maps that are in the axoneme. Not be concerned about us, just to say that they are things that makes microtubules stable. Here we see ciliated cells. So these are the cilia, individual cilium that we see. This is the efferent ducts and the horse epididym, efferent ducts. And you see true ciliated cells and columnar cells, simple columnar cells. So we see the cilia there, and we see the basal bodies in through here. The dark band through there is really the basal bodies below the cell. As I mentioned, one doublet moves in relation to the other doublet, and so as it swings back and forth, you can see that you can even see the singlets at the very end. And it swings the other way, it's the other group, back and forth. Or if you're cutting cross-section, you will see all the 9 doublet microtubules. And whenever scientists first observed that, they come up with the idea that the filaments must be, the doublets must be sliding in relation to one another. That's what happened. In fact, you can remove the membrane and provide ATP there, and you can get these doublets to slide in relation to one another, and they're not confined with the cell membrane anymore. Then they'll just keep on sliding. And also, if you look at a cell, you see that it's not individual cilium that's going there. There's organization in the movement of the cilia, and you can see it's basically in little waves that it moves, and that's very useful for it to be able to like move mucus out in the respiratory system, for example. So there are drugs that influence microtubule assemble. They're inhibitors. Colchicine will inhibit it and destroy it in vitro. Vancomycin also will inhibit assembly. And then you have a stimulator. Taxol will stimulate the assembly. So if you have the tubulin, put Taxol in there, it will make a microtubule associated with it. We thought that you could use some of these inhibitors as a treatment for cancer, but you can't do that because there are cells that are dividing all the time that need microtubules, and they need cell division. Cancer cells are dividing quickly, so why not just target them dividing cells? But in the digestive tract, the lining of the tract is replaced every 4 to 6 days, and so you need to be able to have places in the cells in the body where the cells undergo mitosis at a normal phase. So in summary, microtubules are a cylinder composed of 13 protofilaments, which are parallel, and each of those are composed of individual alpha and beta tubulin. So this is alpha beta tubulin, and they're making the protofilaments, and that's going to make a tube. And also growth occurs like from one end to the other end, and it's the polarity of direction of growth that allows things to be directed in the microtubules. Another cytoskeletal component is the microfilaments, and we see the actin filament here. We have the monomers coming together to make an actin filament. So these are individual actin monomers go together to make an actin filament. This is highly conserved. If you look at a cow's brain and you look at a sea urchin, the actin is very similar. And in plants, actin can be used to direct vesicles along too. Actin associated with proteins, myosin has ATPase activity, and actin and myosin, of course, is important. And tropomyosin is a rod-like protein that causes a linear structure, as we'll see here in a minute. Filament, in contrast, makes a bundle. And so things will bundle. We'll see that right here. If you have filament associated with actin, it forms like a gel. Kind of like, here's what we see. This is the nucleus of a cell, the limits of a cell, and you can see the actin filaments inside there. Kind of like gelatinous stuff for the cell cytoplasm. In contrast, if tropomyosin is associated with it, it makes a linear structure, combines with myosin, and it's actually the striation of the striated muscle. So we see striations in through here, and that is associated with tropomyosin and myosin coming together in linear fashion as opposed to a gel-like fashion in the case of there. So cell cytoplasm, we have the filament associated with it to make the gelatinous material, and also this will allow the cell to stretch and to change its shape without causing any damage to occur. In skeletal muscle, we have the tropomyosin and actin there. We even have the troponin complex that's associated with the trigger to cause the thin filament here, which is the actin, to associate with the myosin in the contraction itself. Here we see a cell just kind of moving around back and forth, but it wants to go in a certain direction. It actually sends the microtubules to the cytoplasm where it wants to advance, and the microtubules kind of push on the cytoplasm there to direct it. And here we can see where we have the actin is pushing on the cytoplasm, and here where we have a centriole, we have the microtubules directing the vesicles to be used as plasma membrane here. So we're pushing along and adding membrane here so the cell can move in this direction. So these are bringing vesicles. That's what they're using microtubules to bring vesicles, but the actin is actually pushing the side of the plasma membrane along, and then these vesicles are adding to the surface. And here we can see it. This is a neutrophil eating up a yeast cell, and here you can see this clear area. It doesn't have other components in there because it's rich with actin, as the actin is pushing the pseudopodia around for phagocytosis to occur. So the function is in cytokinesis, and here we see two cells that nucleus is divided, and now you have the cleavage furrow between the two, and basically what happened is the actin squeezes down on it. Also there's an actin belt around epithelial cells. So support is one of the functions that the actin has. It's usually around the top of a cell, could be the bottom of the cell, but it gives stability to the cell attachments. Here we can see actin in the cell, inside the cell, as well as on the surface of the cell, as we can see. So if we see those again, we can see here, we can see the actin filaments right up next to the cell membrane. Also, here we can see the actin-rich cortex of cells. So right on the outside, next to the plasma membrane, is where you have a rich deposit of microfilaments, because they are actually pushing on the membrane. And then here we see thick and thin filament. The thick filament is the myosin, and then the actin is the thin filament. And how they are aligned, here we see the actin is attached to the Z lines, and then the myosin stays put. So the actin is the light band, and the myosin is the dark band. Also, these microfilaments, or actin, pushes on the plasma membrane of intestinal absorptive cells, and that makes a brush border. So we have uniform microvilli of the same length, basically, of high density, non-branch, that will make a striated brush border, and that we can see here. We see the glycocalyx on the outside, but you see in cross-section the actin. So a microvilli movement and shape. The microvilli can kind of go in and out a little bit, but they don't really beat like cilia do. But you do have a machinery there that can make the microvilli get longer or shorter. It pushes the membrane out, pushes out the cell. And then we have a structural support, microvilli movement, Here we can see this is a cell membrane here, and you can see the terminal web here where the actin filaments actually extend down into the cytoplasm to anchor it for each microvillus to project up to make a striated brush border. And that's what we see here, where the actin's up through there, terminal web in through there. But sometimes the actin can make a sheet, and then that will make projections on the cell or the tissue. Or also the actin can be down here, and that will cause the invagination to occur. So where the actin is located dictates the shape of the structures on the cell. And here we can see in the ear, we have the stereocilia, which is very, very long microvilli is what we see there. There's one cell, and these are the cilia. These are the stereocilia on the surface. And here we can see the stereocilia, very, very long projections, and they have long actin filaments that go not only in the microvillus itself, but also down deep in the cytoplasm. A cell or plasma membrane would be right in through there. So microfilaments function in structure support, movement of cells, change the shape, keep the shape, is what they do. Also, it facilitates movement. It will push the plasma membrane forward so it can attach down here and lose attachment down behind, and that's the way a cell moves. So we have the stress fibers, we have cell cortex, as I mentioned, on the surface, we have the pseudopodia, and the different arrangement of the fibers dictates what type of shape it is, or whether it's a stress fiber to resist things, or whether it's actually pushing the plasma membrane out to maybe have a inspection, a microsphere projecting out of a cell so that it can interact with the environment. Also, the actin I mentioned that was at the top of the cells, and also it can squeeze and change the cell, and you can see how these cells are getting more pyramid shape. Instead of being cuboidal, they're more pyramid shape. And here we see in the embryo where this is making a shape, and it's going to be a structure here. Or you could see a similar type thing in the lens. This is making the lens of the eye right here. And so by having actin filaments tight in through there, like this, it makes these cells more pyramid shape, and that allows it to fold back on itself like this, and to make the lens as we know it today. So microfilaments, contractile proteins, actin and myosin, present in muscle cells. Almost all cells have actin and myosin. Actin is very conserved, and it's just found widely in life forms, animal life forms. And here we see smooth muscle cells, and we have actin in smooth muscle cells. There's also intermediate filaments here. The darker ones are the intermediate filaments. And then when contraction occurs, the actin filaments actually get smaller. Intermediate filament stays the same. And then also we can see the densities on the surface and inside there. Those are the dense structures where you have junctions, attachment of the actin to the intermediate filaments that we have there. And so in summary, microfilaments, contractile proteins, that's what it does. You have a terminal web to anchor the microvilli projecting off the surface, making a brush border. Here we can see where the actin filaments project down deep. They can make mesh. They can exclude other things and other components of the cytoplasm because they make a mesh. And so other components of the cytoplasm can't get up in there. The other filaments are the intermediate filaments, and they are of 5 type, and it is not conserved. It's only appeared more recently. Keratin is insoluble substance of epithelium. And you can put your hand in gasoline, it doesn't dissolve, doesn't burn, because it has a keratin in its surface. And here we see from body wearers where the individual has the keratin is in the epidermis. You see the skin here, which is dermis and epidermis. But also in cytoskeletal components of muscle, we have endesmin is where we have it. Here we see muscle cells for the body world. So desmin is a type of intermediate filament that's in muscle. And then we also have fermentin, which is the for mesenchymal cells. So in the mesenchymal cells and the connective tissue cells, we have fermentin. So this is useful since it's not conserved. Whether you have keratin or desmin will tell you if the cancer is epithelium or muscle or connective tissue. What we have. Also, we have neurofilaments and glial filaments, as well as the 5 classes associated with the nervous system. So intermediate filaments, they use immunofluorescence detection as a tool to determine what type of cell is a cancerous cell. And they go by these 5 different types of intermediate filaments to determine whether or not it is one type of tissue versus another type, and that may lead to a better prognosis and also a better treatment. And then we have myofibrils, which is one myofibril, another myofibril, another myofibril, another myofibril, and they're organized by the intermediate filaments. So when we look at muscle, we see the Z line, the A band, the I band in through there, and how one sarcomere is one filament, myofibril, is associated with another one. The lining up of these, as we see, it's not perfect, but it's pretty good, is due to the intermediate filaments. So intermediate filaments associated with lining up the filaments inside of muscle cells. Intermediate filaments also a structure of epithelial cells in terms of the desmosome. Desmosomes are spot wells, and the hemodesmosome is a half of a well because it's connective tissue below, or collagen substance, extracellular substance below. And so here we can see desmosomes is what holds cells together. Hemodesmosomes is at the bottom of the cell, and here we see collagen. So intermediate filaments also function. It's extracellular component you see in nails, you see in feathers, scales is what we see. Now, intermediate filaments are different types, as we mentioned to you. There's a basic component which is common among them, and then what's different is the amino terminus and the carboxyl terminus for the different types of intermediate filaments that are located in the different tissues of the body. And so here we can see the overlapping of these units to make the intermediate filaments as we see here and here. And so of the cell, we have the different type of cytoskeletal components. The actin, we see the actin all around the cell, or it's associated with the cell membrane projecting up in the microvilli. The microtubules are coming from the centriole, radiating in different places, and intermediate filaments are attaching the desmosomes, or the spot attachment between adjacent cells, as well as the hemodesmosome. So in review, cytoskeletal components are really part of the non-membranous components. We have microtubules, which direct vesicles. We have intermediate filaments, which hold muscle, myofibrils together, as well as other functions, stabilize cells. And also we have microfilaments that help cells move to direct the movement of cells. Microtubules associated with chromosomes pulling apart. So next time we will talk about the nucleus and mitosis, or we'll go to Oregon and see Mount St. Helen. This is from a plane when we flew over to that area, and there it is. If you look, you can see our chairlift. Right up through there is a chairlift going up through there. There was snowing, and there was snow up there, and there was skiing in the summertime. Also, this is a view of the Pacific. The sun is going down in the Pacific, and this is the bridge between Washington State and Oregon that we can see. We want to thank many books, because these notes were taken from books. I was not the original source of these pictures. We want to thank the various books, as well as a couple things I got from the internet as well. I just want to acknowledge that they contributed those. I did not produce those myself, and for those we say many thanks for us to use for educational purposes. Thank you, and if this is useful, please share the website, peertamu.edu, and also please share the YouTube channel, VIBS Histology, with your friends and neighbors. Take care. Bye-bye.